Here we are back with lecture 16. We are going to begin by talking about four major sources of genetic diversity. So you should be there on your lecture outline right now, just above 22.2 on your outline. And with these four major sources of genetic diversity, the question that we're addressing here is how can any two given parents produce such an incredibly varied genetic group of offspring? If you think about it, then certainly you're genetically different, assuming that you're not an identical twin. You're genetically different than any of your siblings. You're, of course, genetically different from your parents. And don't write all this down, by the way, but just think about it. So you're different than your parents in terms of genetics, in terms of the genes you carry and the information that those genes carry. So you're different than your siblings. You are different than anyone else in your family of this generation. So cousins and so on, you're different than any of them. You are different, well, we could keep going, but essentially you're different genetically, almost certainly, than anyone else that's living on the planet today. And you are also almost certainly genetically different than anyone who has ever lived on this planet before. So again, the question we're addressing here, how does this diversity come about? How can two given parents produce an almost endless variety of offspring? And you'll notice now on your lecture outline, I've jotted down for you off to the right those four sources of genetic diversity. Three of these, you may recall, were addressed in the two videos that you guys watched, having to do with reproductive systems, having to do with meiosis, actually. And the fourth we'll talk about also. So first of all, this process of crossing over. I think at this point, you guys are probably able to not take a whole lot more notes and just to kind of sit back and think about each of these as we go through them. So crossing over, remember this process of two homologous chromosomes when they synapse during prophase 1, these two homologous chromosomes trading pieces of one another. And on each of these pieces, there will be a number of individual genes. And what's happening, remember, with this process of crossing over is that now we get different combinations of genes than we could otherwise have. And so one of a pair of homologous chromosomes has now traded a piece of itself for a corresponding piece that carries some new genes. This is only this one pair of homologous chromosomes that we're talking about in this one particular round of meiosis. So we're just talking about starting with one individual cell, and that cell, through the process of crossing over, is going to, again, each of those homologous chromosomes in that cell during the process of crossing over is going to acquire a new combination of genes than it might have otherwise had. Again, it just refers to, it just applies to this one cell producing four gametes, producing four sperm cells or four egg cells. We have another cell, if you can picture this now, a different cell also undergoing meiosis and producing four gametes, producing potentially four, depends on the sex, but producing potentially four gametes through meiosis 1 and meiosis 2. And again, here in this second cell, different pieces of homologous chromosomes are likely to be exchanged. Different chunks are traded compared to the chunks in that first pair of homologous chromosomes that were traded. And so we have a new combination here just with this one particular pair of cells. And again, we've been talking for the moment just about one pair of homologous chromosomes. But remember, in humans, there are 23 pairs of chromosomes, and 22 pairs are entirely homologous. And so somewhere else along the line of homologous chromosomes that are lined up during metaphase 1, somewhere else along the line, another pair of homologous chromosomes will be trading pieces. And in a second cell, again, that same pair of homologous chromosomes will be trading different pieces. So now we have even more genetic diversity. And we can just expand this idea. And so that third pair of homologous chromosomes in cell number 1, if you will, they exchange pieces. And this results in new combinations of genes that we couldn't otherwise have. And then in cell number 2, this third pair of homologous chromosomes, they're most likely trading different pieces. And so what we end up with, just in these 2 cells that we're starting with, are many, almost unlimited possible combinations of genes due to various possibilities of crossovers, various combinations of crossovers that we get. And again, this is just in 2 cells that we've been talking about. Extend this out to many cells, and you get many different genetic variations in sperm cells that are being produced. And so again, crossing over, even if only one crossover per pair of homologous chromosomes occurred during meiosis, you'd get just an unbelievable number in terms of genetics of different sperm cells or different egg cells being produced by, again, one cell that we started with versus a second cell that we started with versus a third and a fourth, and on infinitum. To make this even more impressive, typically there, and again, I'm still suggesting you not take notes, so just kind of sit back and listen and consider this. So furthermore then, typically any one pair of homologous chromosomes will not just trade one chunk with one another, but typically each pair of homologous chromosomes will trade several different chunks with one another. And again, the chunks that are traded in cell number 1 that we're starting with and which is undergoing meiosis, these chunks are traded completely independently from the chunks that are traded in cell number 2 versus the chunks that are traded in cell number 3, and so on. And so again, just an astounding number of different types of sperm cells and egg cells that are produced just by this process of crossing over. Number 2 on your list then, and again, we're talking about 4 major sources of genetic diversity. Again, the question that we're asking is, how is it that any given pair of parents can produce such an incredible diversity, genetic diversity in terms of their offspring? And the second source of genetic diversity now is the independent assortment or the random shuffling of chromosomes. And this occurs during metaphase 1. This is much more easily explained and much more easily absorbed with a visual and particularly with a video. And so I would just direct you back to one of those videos, one of those 2 videos on meiosis. And at the end of this process of independent assortment of chromosomes, the point was made that each of us, each individual person, since we have 23 pairs of chromosomes, each of us can then produce 2 to the 23rd unique genetic types of gametes, of sperm cells and egg cells. And so the point was made again, each of us can make more than 1 million different unique types, genetically speaking, of gametes, just through this process of the independent assortment of chromosomes. And it's actually many more than 1 million. So you and I and each of your classmates can make all of these different genetic types of sperm and egg cells just due to the independent assortment of chromosomes during metaphase 1, due to the fact that when the first pair of chromosomes begins to make that line, those two lines of 23 pairs of homologous chromosomes, the second pair of homologous chromosomes come in and orient themselves underneath, if you will, the first pair independently, regardless of the position of the first pair. And then now picture that third pair of homologous chromosomes coming in and joining the lines, if you will, as far as which is on the, left side, which is on the right side, independently of what happened with the two pairs of homologous chromosomes above. And so again, just a tremendous number of different possibilities of genetic types of gametes that we can produce, sperm and egg cells we can produce, just by this independent assortment of chromosomes. Number 3 then, we have random fertilization. And one of the videos, and I jotted down the quote, what we're talking about here is the random pairing of gametes during sexual reproduction. And again, I would say just instead of jotting down that, what you might just want to jot down for random fertilization is that any given type of egg cell is equally likely to be fertilized by any given type of sperm cell. Any given genetic type of an egg cell, again, is equally likely, any given genetic type of egg cell is equally likely to be fertilized by any given genetic type of sperm cell. So again, if you have that down in your notes now, just sit back and think about it. What are we saying? Each month or so, a female will produce and release typically a single egg cell. And that single egg cell can be, each 28 days, each month or so, that single egg cell that's released can be one of over 1 million different types of egg cells that woman is able to produce. And at the same time then, each male is able to produce well over 1 million different genetic types of sperm cells, as we said. And now we're saying random fertilization, whatever type of egg cell was released by the female at this particular time, it's equally likely to be fertilized by any one of more than 1 million different types of sperm cells. And as a result of this, and you don't have to jot down the number, but just as a result of random fertilization, there are more, for each given couple, for each given male and female, there are more than 8 trillion different genetic combinations just by any type of sperm cell fertilizing any type of egg cell. And then finally, we have mutation for the 4th major source of genetic diversity. And here you can jot down in your notes simply the fact that mutation is a change in structure of a DNA molecule. And recall that chromosomes essentially are DNA molecules. And so now, due to mutation, this makes even many, many more possible types of possible genetic types of offspring possible. And we'll stop here with that for lecture 16.